Osmosis. Osmosis is the process that involves the movement of water molecules from a region of high water concentration to a region of low water concentration across a semipermeable membrane. It is a special type of diffusion that involves water molecules moving across a semipermeable membrane. The process of osmosis can be demonstrated by placing a whisking tubing, half filled with sugar solution, in a beaker with distilled water. A whisking tubing is a commercial synthetic transparent semipermeable membrane. It represents the cell membrane in living cells. The whisking tubing contains both water and sugar molecules. Since the beaker contains only water molecules, it therefore contains a higher concentration of water molecules than the whisking tubing. The whisking tubing becomes swollen due to increase in its contents. This is because the water molecules will diffuse from a higher concentration to a lower concentration, that is, into the whisking tubing, and thereby increasing its contents. The wall of the whisking tubing allows only the small water molecules to pass through, but not the larger sugar molecules. The membrane is, therefore, said to be semipermeable. The process of osmosis can also be demonstrated using two solutions of different concentrations. In this case, water molecules move from the dilute solution to the more concentrated solution. The movement of water molecules continues until equilibrium is established. At this point, the two solutions are of the same concentration and are said to be isotonic. Therefore, osmosis is also defined as the process whereby the solvent, water, molecules move from dilute solution to a highly concentrated solution across a semipermeable membrane. A dilute solution is described as hypotonic because it has less solute molecules, while a highly concentrated solution is described as hypertonic because it has more of the solute molecules. Osmotic pressure. When a concentrated solution is separated from distilled water by a semipermeable membrane, the solution develops a force with which it draws water through the semipermeable membrane from the distilled water. On the other hand, pressure can be applied against the semipermeable membrane to stop water molecules from the distilled water entering the concentrated solution, or to stopping osmosis from taking place. This pressure is referred to as osmotic pressure. The osmotic pressure of a solution is measured by an instrument called osmometer. Osmotic potential. Osmotic potential is the hidden pressure of a concentrated solution that only becomes real when the solution is separated from distilled water by a semipermeable membrane. It is a measure of the pressure a solution would develop to draw water molecules from pure water when separated by a semi-permeable membrane. Water relations in animals. The membrane of animal cells is semipermeable, just like the whisking tubing or cellophane. The cytoplasm contains dissolved salts and sugars in solution form. If an animal cell, for example, a red blood cell, is placed in distilled water or hypotonic solution, there would be a net flow of water into the cell by osmosis. The cell would swell up and eventually burst. The bursting of a red blood cell is known as haemolysis. If a similar red blood cell is placed in a hypertonic solution, then water would be drawn out of the cell by osmosis. The cells would shrink, a process called crenation. This behavior of red blood cells placed in various concentrations of solution indicates that body fluids and the blood plasma surrounding the cells must be kept at the same concentration, i.e. isotonic, as that which is found inside the cells. If this is not done, the cells will either gain water and burst or lose water and shrink, and their functions will be impaired. However, the body has a mechanism of keeping these concentrations constant within a narrow range. Water relations in plants. A plant cell has both a cellulose cell wall and a cell membrane. The center of the cell contains a vacuole with sap. The sap is a solution of salts and sugar and is bound by a membrane called tonoplast. The cell membrane and tonoplast are semipermeable, while the cellulose cell wall is fully permeable, thus allowing solutes and water to pass through freely. If a plant cell is placed in water or hypotonic solution, the water will move into the cell by osmosis and cause the cell wall to distend. The cellulose cell wall is rigid and does not allow cells to burst as in animal cells. As the cell gains more water, its vacuole enlarges and exerts an outward pressure on the cell wall called turgor pressure. This pressure increases as more water is taken into the vacuole, which pushes the cytoplasm against the cell wall until the cell wall cannot stretch anymore. The cell becomes firm or rigid and is said to be turgid. When the cell wall is being stretched towards the outside, it develops a resistant pressure to stretching that is equal and opposite to turgor pressure, called wall pressure. When a plant cell is placed in hypertonic solution, water molecules move out of the cell into the solution by osmosis. As the water moves out the cell membrane pulls away from the cell wall and the cell becomes plasmolyzed. The process by which the cell membrane shrinks and pulls away from the cell when a plant cell is placed in a hypertonic solution is referred to as plasmolysis. When the cells of a tissue get plasmolyzed, the tissue becomes soft and less rigid, and the tissue is said to be flaccid. Plasmolysis can be reversed when a flaccid cell is placed in distilled water, and this process is called deplasmolysis. Wilting. Plants lose water to the atmosphere through transpiration and evaporation. The cells in the plants lose water and draw more from the soil. It reaches a time when the rate of water loss to the atmosphere is more than that of absorption from the soil. Turgor in cells is reduced. The cell wall loses its rigidity. The cells shrink and the plant droops. The phenomenon is observed in plants on hot and dry days and is called wilting. At night, plants recover from wilting because their stomata are closed and rates of both evaporation and transpiration are reduced. However, if water supply from the soil is inadequate, the plants do not recover from wilting and are said to have undergone permanent wilting. Factors affecting osmosis. Concentration of solutions and concentration gradient. Osmosis is greater in solutions that have greater differences in osmotic pressure. The greater the concentration gradient between two solutions that are separated by a semipermeable membrane, the higher the rate of osmosis. Temperature. Low temperature slows down osmosis. Increase in temperature causes an increase in the rate of osmosis. Extremely high temperature destroys the structure of the cell membrane, hence hindering osmosis. Role of osmosis in organisms. Absorbing water from the soil. Root hair cells of a plant absorb water from the soil by osmosis. Osmosis also facilitates for water distribution from cell to cell in the body. Support. Water taken into cells increases cell turgor, hence, cells become firm or rigid and therefore gain support. This type of support is important in seedlings, leaves and herbaceous plants which are less woody. Opening and closing of stomata. The guard cells synthesize glucose through the process of photosynthesis in the presence of light. As glucose accumulates in the guard cells, their osmotic pressure increases and this enables them to draw water from adjacent cells by osmosis. This action results in the guard cells becoming turgid, causing the opening of the stomata. The opening of the stomata allows gaseous exchange to take place. At night the guard cell cannot photosynthesize and therefore, their glucose levels go down, leading to a decrease in osmotic pressure. The guard cells, therefore, lose turgidity and hence the stomata closes. Feeding in insectivorous plants. These are plants that trap insects using special structures that suddenly change their turgor pressure when disturbed. The change in turgor pressure enables the special structures such as leaves to trapping insects, which are then digested to provide nitrates. Osmoregulation. In kidney tubules of animals, water is withdrawn from the tubules through the tubular walls by osmosis. The water finds its way into the surrounding blood capillaries. This helps the animal to regulate its osmotic pressure. Thank you for watching this biology lesson. Please subscribe to this channel and other Genius channels relevant to your level of study.